The functions of the histones is to form the nucleosome with the DNA, helping in the transcription of the DNA and all the process. The gene of the histone H4 is located in the chromosome 12 in humans, in lot CP 12.3, but in some animals like the mouse is located in the chromosome 3 in lot CQ 30.30. Histone H4 forms the nucleosome, but to be functional it has to form a complex with histone H2A, H2B, H3, H4 and with H1 to close the nucleosome, as we see. It has a pair for each histones, but except the H1. So the nucleosome has to contain chains of all these histones apart from the one of H4. Histone H3 and H4 bind to form a H3-H4 dimer, and histone H2A is joined with the histone H2B. Then they join all together as we see, and the linker DNA surrounds all the histones. Then histone H1 closes the linker DNA and the nucleosome is formed. Histone's structure is formed by a 3A helices connected by 2 loops, and H4 histones protein is formed by 102 to 135 amino acids, which shares a structural motif. The gene will translate into RNA and make its proteins. The gene always do a process called transcription and translation, consisting in a series of processes. Transcription is the process in which the DNA is used like a template strand to make the RNA, and translation uses the information of the RNA to make a polypeptide protein. Transcription takes place in the nucleus, but translation takes place in the cytoplasm of the cell. For transcription, you need to have a point to start transcripting in the template DNA strand. So this is called the promoter region. The point when you have to finish translation is called terminator region. In transcription, when the template strand of the DNA is used to make the RNA, its structure changes the letter T into U. In DNA, you have the nitrogenous bases that change, and this makes the DNA. Each nitrogenous base has the complementary base pair. So in transcription, you use the complementary one to obtain the RNA. But you must change the letter T in a U like we see it. Then, when this process is finished, the RNA has some parts that are not coding regions called introns, and the coding regions called exons. For translation, these not coding regions have to be removed, so spliceosome is required to just remove introns and the RNA is prepared to begin translation. In the mRNA, its 3 nitrogenous base is called a codon, and each codon is used to determine which amino acids are selected. Then all amino acids are joined to form a protein. All cells in our body express H4 histone gene. They are essential. The histones are located in the nucleosome, so in the nucleosome of the cell, the nucleus of the cell. This means that this protein is really important. We can't live without this protein, so if this protein doesn't work, we are in a big problem. If the histones doesn't work, the nucleosome doesn't exist, so the chromosome either and you won't be existing, because without any chromosome you can live or develop. There are no mutation with these proteins or any disease, because you need to live, so if this protein stops, you die. But there are some modifications called post-traductional modification, which just regulate the functions of histones.